product of cell division. So if you would turn to 163, we're on section 9.3 talking about cancer and the cell cycle.
93?
163, section 9.3. Let's take a look at this real quick. This shows the difference between how plants and animal cells do cytokinesis.
Cytokinesis is the phase of the cell cycle during which the cytoplasm actually divides. It follows nuclear division and involves the splitting of the cell into two cells. In animal cells, cytokinesis is achieved by a constricting belt of actin filaments interacting with myosin. As the filaments slide past one another, the diameter of the belt decreases, pinching the cell and creating a cleavage furrow around the cell's circumference. Constriction proceeds and the furrow deepens until it eventually slices all the way into the center of the cell. At this point, the cell divides in two. In plant cells, membrane components are assembled in the center of the cell to form the phragmoplast. These components are vesicles derived from the Golgi apparatus. The vesicles travel along microtubules, and their content forms the cell plate. The cell plate continues to grow outward until it reaches the cell membrane along the perimeter of the cell. Cellulose contained in the vesicles is then laid down between the new membranes, creating two new cell walls. Pectin is also deposited in the space between the daughter cells.
So a plant cell doesn't pinch in like an animal cell does. You see, the animal cell pinches in to divide. But the plant cell just kind of builds a new wall there in between the two.
Let's bring down this wall.
That's because plant cell walls are tough and are hard, and they don't bend. Yeah, there we go. See the way the plant does it?
Just creates more stuff.
Yeah, it creates new cell wall. It builds a new cell wall, basically, between the two cells.
That causes them to split?
Yeah.
But otherwise, they follow the same steps before that?
Yeah, they follow the same steps before that. I think I have some video footage of it here. Actual video footage. Yeah.
The new cell wall can be seen forming as many vesicles fuse along the center of the cell. Two new animal cells
Watch this cell right here.
as the cytoplasm pinches off between the nuclei.
See it pinch?
That's an animal.
That's an animal. Yep.
That was so cool.
Very nice, huh?
Can we watch that? Or is it not that good?
We just watched it.
Move it on a microscope.
Oh, on our microscope? Yeah, that's hard to do. That's some good microscopy. You could probably see it if you worked at it. But that sounds like a graduate school science project for microscopists. Okay, so if you're interested in cloning techniques and such, it talks about it. There's a special reading section on cloning on page 162. I don't know if you know this, but they've cloned sheep before. You can take a cell from a sheep and clone it and make a new sheep that's just like the original sheep. And they've done this with not only sheep, but rats and dogs and cats and mice.
Why haven't they done it with people yet?
It's illegal to do with people. They don't want anyone experimenting on people, because 99% of the time, 95% of the time, the cloned animal dies in some way during development. It's only a small percentage of the time that's successful. So they don't want to do that with people. Why is that? I don't know. A lot of reasons. They're just not very good at it.
Would the clone like feel
uncontrollably and will not stop dividing. Jessica, you with me?
Yes.
And the cell just multiplies and multiplies and multiplies and doesn't stop. And all it takes is one cell to do this. If you have one single cell, let's say it's on your hand, a little microscopic cell, and it starts dividing and won't stop, well, suddenly you'll have 2 cells, and then 4, and then 8. Each one of them a copy of the one before, and each one of them dividing uncontrollably. 2 cells, 4, 8, 16, 32, 64, 128, 256, 512, 1024, 2048, 4096, 8192, 16,384. Yeah, I was on the state championship math team. And pretty soon you got a big lump of cells. You know what they call that?
A tumor.
A tumor. And that can be dangerous because cells can break off from the tumor and start spreading around your body. And these are cells that are multiplying out of control. And one of those cells could end up in your brain or in your heart, in blood vessels in your heart, and multiply there and form tumors there and block up your blood vessels. Or squeeze your brain so that the cells die. And that can kill you. So all it takes is one cell to do this, and you're in some trouble.
Is apoptosis cell suicide? Yeah.
Yes. Here's normal cells are differentiated. That means they look different. Muscle cells look different from bone cells, look different from nerve cells. But cancer cells are usually non-differentiated. They all the cells look the same. And if you're a pathologist, you go to school for about 10 years, and you look at these cells over and over under the microscope, and you can spot them. A good pathologist can spot a cancer cell amongst a bunch of other cells just by looking.
I have a pathologist friend.
Yeah?
Yeah, he actually worked at McDonald's and then he quit and now he's a pathologist because he was in school.
What's his name? Is he from here?
Yeah, because I used to get a Diet Coke from every single day.
Is he at our hospital?
Yeah, I think so.
Huh.
His name is...
Dr. Hanley?
No.
Oh well. We're going to go, after the AP exam, we're going to try and go to the hospital and look at the pathology lab. Cancer cells have abnormal nuclei, and normal cells have normal-looking nuclei. Cancer cells won't kill themselves. Do not undergo apoptosis, but normal cells will. The cancer cells don't have contact inhibition. That means when they're touching each other, normal cells, when they're touching each other, won't grow anymore. But cancer cells, when they're touching each other, they'll grow right on top of each other. Cancer cells are disorganized and multi-layered and undergo metastasis. That's a word meaning to spread. To get this way, a cancer cell has a bunch of mutations. Mistakes in the DNA that have consequences that cause it to be this way. This is why you usually see cancer in old people, because it takes a while to build up all those mutations. If you're really unlucky, you can build up all those mutations fast. And some people are more genetically predisposed to getting cancer than others. But cancer is pretty rare amongst young people. It's pretty tragic when a young person gets cancer, because you just don't see that very often. But it can happen. You just have to be unlucky.
Does that mean that cancer could be, like, the replication of cells could be happening in us right now, but the mutations aren't going to show up until we're old?
The mutations can be happening right now. But the mutations you have to have many mutations to develop cancer. So it might eventually show up. Yeah, it's happening in all of us right now. And some people, it shows up later than others. Some people, they'll die of something else before they ever get cancer. Over half the people end up getting some sort of cancer at some point, and so half of us in this room will have to deal with it. It's not always deadly. And they're getting better and better at curing it.
Is that benign?
Yeah, it can be benign or malignant. Benign means it's just a growth and you can cut it off and go about your life. Malignant means it's likely to spread or has spread around your body. So you don't want to hear about a malignancy. That's no good. So here's a cell that's become cancerous. Pretty soon it will develop into a lump, a tumor. And here's this cell has mutated even further where it's likely to spread. You usually want to cut off a lump, a tumor, because even if it's not likely to spread, you could get further mutations in one of them that allow it to spread. And if this one gets a mutation that allows it to spread, it'll break away from the others. And spread down deep into the body. And here it spread into lymph vessels and blood vessels, which are vessels down deep in your skin. And we'll get in those vessels.
Is that like lymph nodes?
Yes, lymph nodes. And travel through that vessel to some other place in your body and then start multiplying there. One of the guys I played golf with died from cancer, and he said he had a little cancer on his skin and they cut it off, and about 4 months later he had some problems and he went to the doctor and they did an x-ray of his body and he had thousands of tumors everywhere in his body. The doctor basically said, "You got 2 months or whatever." And I think he was dead in like 3 weeks. It's terrible. But what can you do at this point? Well, once a cancer starts to spread, you can try what's called chemotherapy or radiation therapy, in which they just hit your entire body with a bunch of radiation and chemicals. The thing about cancer cells is they're usually pretty weak cells, and they die pretty easily. You wouldn't think that about cancer. You'd think those cells must be very strong. They're not. They'll die pretty easy. So if you hit your whole body with stuff that kills cells, hopefully it'll kill the cancer cells before it kills all your regular cells. And you can kill all the cancer cells that way.
Does that work?
Yeah, sometimes chemotherapy works. And the person recovers and all the cancer cells are dead and they go on about their lives. Sometimes the cancer is so progressed where all that radiation will kill too many of your normal cells before it'll kill all the cancer cells. And you can just die from the chemotherapy. Sometimes it's so progressed the doctor knows the chemotherapy won't ever kill it all and they don't even prescribe it. They just say you got a few months, enjoy them, and you're not going to make it very much longer. So yeah, cancer can be a pretty sad thing, and they're coming up with better and better ways to treat it. You know, when I was a kid, there were a lot of cancers that were just untreatable, and now most cancers are treatable to an extent. You got to catch them early. That's why it's good to go. If you ever see something on your body, it's a lump or something that doesn't look right, you go to the doctor. Don't just let it sit there. All cells have two types of genes. And the two types of genes are called proto-oncogenes and tumor suppressor genes. And this picture's in your book on 165, and it talks about it on 164. You write these words down. These are words you need to know. Proto-oncogenes. Proto-oncogenes, I drew it in green there because they're like the green light. They're genes that tell cells to divide. These genes make proteins that are used in cell division. And other things that cause cell division to happen. So that's why it's colored green. It's like stepping on the gas for a cell. It causes division. Now there's another type called tumor suppressor genes. And that's like the brake for a cell. That stops cell division. These tumor suppressor genes make proteins and such that slow down cell division or stop it. Do you remember those little stop signs? that were present in the cell cycle. Let me back it up a little
G1 and
bit.
G2.
Yeah. All these little stop signs.
A long way back.
God, it's a long way back. I might not should have done this. I didn't realize I was in for such a... Oh my goodness.
You know, you need two projectors to really see how.
Oh, here we go.
Yeah.
See, remember the G1, G2, and M checkpoints? Those are actually proteins that can stick to the DNA or some other way stop cell division. And we don't want to get into all the mechanisms of exactly how it works because it's very detailed. But it's the tumor suppressor genes that make the proteins that stop the cell cycle. Yes?
Apoptosis is cell division.
Apoptosis is cells killing themselves, cell suicide.
Well,
Thumb exercise.
Let's go all the way back. I know there's an easier way to do this, so I'll do it. I like to click.
no, it's a cool vision. Weird.
It's pretty cool. Mouse do it like floats in the air. How it works.
Yeah, it's called an air mouse. So these tumor suppressor genes stop the cell cycle, and proto-oncogenes get the cell cycle going. So if there is a mutation, if there is a screw-up in the tumor suppressor genes, the cell cycle could just continue without stopping, you see? And that could lead to cancer. If there's a mutation in the proto-oncogenes that make them more active than they should be, that can also lead to cancer. And it's usually a combination of both of them that end up causing cancer. Extra proto-oncogene activity and extra and less tumor suppressor activity. Both of those can cause cancer.
During the first phase of the cell cycle, G1, there is a chemical checkpoint that a cell must pass to begin the process that leads to cell division. The protein products of several tumor suppressor genes act at that checkpoint to control cell division. Cells with mutated forms of the tumor suppressor genes may not be able to stop the cell cycle at the G1 check.
This kind of shows how all this works. And it's a little bit above what we're worried about right now. This shows a cancer spreading. This is a pretty good video.
A cancer begins with the transformation of a single cell, enabling it to divide without limitation, forming a primary tumor. Metastasis is the formation of secondary tumors at other sites in the body. Cells break away from the primary tumor, passing through the basal lamina and entering the blood or lymph. When a breakaway cell establishes itself elsewhere in the body, it can divide and produce a secondary tumor.
And a lot of times these cancers will spread inside your body without you feeling it. They don't cause any pain. Because you don't have a lot of pain sensors inside your body. So it can get pretty well developed before it hits something that causes a lot of pain or problems. So this is why women should check themselves for breast cancer and guys should check themselves for prostate cancer. Those are the two most common types of cancers. And do that regularly once you get to a certain age. At your age, you probably don't have to worry about it as much, but when you get 30, 40, 50 years old, it's important.
Why do people say, oh, don't drink the water bottle that's been sitting in your car and you can get cancer? Like, what is, it's still like
Leaching
heated
chemicals.
plastic.
Oh, the heated plastic will melt into
there's-
it. I don't know if that's a proven thing.
There's so many things like that.
Well, I mean, there's
Yeah,
things
every
that
day
cause
there's
cancer,
something new
like
that
the
says,
sun
"Oh,
and
cancer."
chemicals, right?
Yeah, the sun can cause it. Chemicals can cause it. You don't want to get too many weird chemicals in your body, and that's what the heated plastic is.
Okay.
I don't know if that really causes cancer. You hear a lot of things that aren't actually true, but it might. That's a big breast lump.
When you're diagnosed with cancer, do they tell you what it is? Because I feel like most people who get it don't even know what's going on.
Well, this is why you need to learn this stuff. Because they might tell you, and you have no idea what it means. So you need to learn what these words mean. If they tell you you have a malignancy, if you hadn't studied this stuff, you won't know what that means. But this is an endoscopy. This is kind of a video showing sticking a scope up a guy's butt, or a girl's, and they're looking for tumors. And, oh, they found one there. Look at that. There's a tumor there inside your colon, and that could be a colon cancer. And what they'll usually do is they stick a little clipper up there, and they clip off some of it, and they do what's called